Thank you very much for your kind introduction. And good morning, everybody. I hope that you do see my presentation.
Yes, we can.
Okay. Okay, very good. I really would like to start by thanking. Just a minute. Trying to click. Oh, here. Okay. So I'm from Al-Quds University, as already said by the nice lady. And Al-Quds University, the second biggest university in Palestine, and it is located at the eastern side of Jerusalem. So hopefully after this pandemic is over, we will be able to see each other, not virtually, but also here in Jerusalem. So please all feel invited. I'd like really to start with acknowledgement. Usually people finish with that, and I want to acknowledge Professor Hu Long Ng, who initiated a group of researchers, a cluster actually, of researchers worldwide, and of course, Assistant Professor Abraham Pile Karlapoti. I'm sorry if I make it wrong, for really his very cooperative and bringing this idea almost a month ago and bringing it to reality within a month. And of course, I would like to thank all the members of the organizing and scientific committee for the very cooperative and very fruitful work. This is the outline of my lecture. Actually, Professor Dobley gave a very kind introduction and in a way is very complementary to what I'm going to talk about. So my talk is going to be divided generally into two halves. One is an overview of what's going on, what are most promising targets in terms of a medicinal chemistry perspective. And I hope that time will allow me to also talk a little bit about the efforts we are doing together since something like 3 months, mostly virtually and in silico, in order to identify some lead or hits that could be antiviral drugs in the future. So we're talking about a very early stage, really, of identifying novel compounds. Well, coronavirus and COVID-19 turned to be a very challenging pandemic. I was just looking last night until 11:30 p.m., so a little bit before midnight. There has been, according to the BBC, there has been more than 10 million people who have been infected by this virus and more than 5 million of deaths. In Palestine, actually, there has been, we are also part of this world, and we are infected, and the virus is here like everywhere. The first cases were in the first week of last March, something like 4 months ago, actually a little bit more than 3 months, yeah. And now we are hit by what is defined as a second wave. So we are around 5 million Palestinians, inhabitants altogether here. And we already have more than 3,100 confirmed cases, 11 deaths. What is alarming actually is that since last week, we are counting numbers in one day. For example, 300 cases, which is more than the 1 month of March. So it's a bit alarming. And I think we all, as scientists worldwide, have to join efforts in order to find some kind of treatment that can help patients worldwide and, of course, contribute to the humanity in this plan. The current situation is there is no effective vaccine until now. We are hoping to get it within a year or so. There is no one approved drug that was designed for COVID-19. There, as Professor Corby told, there are repositioned or repurposed drugs with certain limitations. What we know biochemically about the critical events and steps in the viral life, the replication cycle, we now know really much more than 3 months ago or half a year ago. And it's believed, as already said, that this SARS-CoV-2 is a mutant that emerged out of SARS-CoV-1, which again can be helpful in terms of medicinal chemistry efforts to identify drugs. From an evolutionary standpoint, it's not good for a virus to be fatal for the host. And SARS the first, SARS-CoV-1 in 2003 was particularly lethal, but wasn't able to spread so quickly like SARS-CoV-2. This is a hint that I really want everyone to think about it, taking in consideration what's happening with this pandemic. We can't talk about targeting the virus without actually hitting a little bit shortly the life cycle or the replication or duplication cycle. Already it has been said that spike protein recognizing the angiotensin converting enzyme 2, and this recognition is very essential, recognition and binding for the internalization of the virus to the host cell. And one of our efforts actually is aimed against targeting this recognition between spike and the ACE2. I will come to that later. Of course, later on, after the genomic material of the virus is translated by the host machinery to the polypeptide 1A and polypeptide 1AB, came the so-called the role of the proteases, 3CL-Pro, which is called the main protease, and the papain-like protease, PL-Pro, in order, and they play a very critical role. That's why more focus and effort worldwide is now put on those two proteases. And I will give an example also in a bit more details on remdesivir and the role of medicinal chemistry in repurposing of already known drug into as a treatment or potential treatment for SARS-CoV-2 or COVID-19 pandemic. So my focus will be on those three parts, but of course, every single step molecularly, biochemically, is, as already said, is potentially a viable target for the virus. It's known that drugs can have many resources. Of course, synthetic products coming from synthetic labs, medicinal chemistry labs, organic medicinal chemistry labs, or bioinorganic medicinal chemistry labs. Unfortunately, this is a very scarce resource right now because most of the labs worldwide are closed due to the lockdown, including my own lab. Natural products, again, people are trying non-natural products. Virtual screening and structure-based molecular docking studies are now really getting more and more attention. And the combination of two, because to say it's easy to do if the experimental labs are limited those days. High focus has been put on drug repurposing. Out of that, we want to come with an inhibitor that is either a peptidomimic or better a small molecule, but a peptidomimic is also fine with a drug-like properties, of course, and we hope to get some kind of structure-activity relationship and to understand the mechanism of action of each drug. So if we have a already known drug, that will be very helpful to have those details later on. And potential molecular targets, generally speaking, are in the context of COVID-19 are divided into two types. One is the host-based targets, And I will mention that, and I will direct the audience and the participants to a very excellent review that was published very recently, but I will not touch but mention, and the virus-based targets on which I'll give a little bit more information and exploration. Host-based targets are the targets that are already existing in the potentially infected cell. So say the cell in the lung, what is there? And one of them is, of course, the ACE2. The other is the cleaving enzymes, already has been mentioned by the previous lecturer wonderfully. And of course, the DDP4, we are speaking about the aminopeptidase. And of course, if anyone is interested, this is a very kind and really explorative review about the host-based targets. A new trend is coming, which is called the host network. And people are trying to, and there are very good works coming out of that, are trying to understand what proteins are needed for the cell, for the division and replication of the virus, and to target them in order to alleviate the viral infection. Targeting, again, the entry of the virus is very important. And these are just a very few compounds. I will not comment much of them, but this is a list of 11 molecular targets that people worldwide are trying to target them in order to prevent or affect or reduce the entry of the virus to the host cell, which is very important, by the way. Virus-based targets, in which I will comment a little bit with more details. Of course, it has been already explained very well how spike protein, virus-based targets are divided into two types. The first subfamily is the structural proteins, and the second subfamily is non-structural proteins. Structural proteins are spike protein, envelope protein, membrane, and nucleocapsid. And it has been already explained wonderfully what is the role of the spike protein. I'll touch into that a little bit in depth, because I'm together with my partners, we are tackling this protein in order to prevent its binding, hopefully, to the ACE2 inhibitor. Doing so, we hope to end up with a molecule or molecules that can prevent the entry and the infection ability of the virus to the host cell. And I will, of course, comment a little bit more on the proteases, on the main protease, and a little bit on the RNA-dependent polymerase, again, from a medicinal chemistry point of view. But there are 16 non-structural proteins that are well characterized, and for many of them, there are already extra structure reported in the protein data bank, as shown in this slide. Structural proteins, again, what is important in this context of this presentation is the spike protein, because it is actually the gate opening, in a way, of the host cell that is going to be infected by the virus. And without those spike protein, it is believed that the virus cannot enter, cannot be internalized into the cell. That's why targeting this protein that's decorating the outer envelope of the virus is very critical in a way. And I'm not going to repeat what has been already mentioned and explained very wonderfully by Professor Bobley. It's known that the S1 protein contains what is defined as the receptor binding domain that recognizes the extracellular peptidase on the ACE2. Then there's steps of cleavage that help in changing the configuration of the spike protein and help it actually, in a way, it catalyzes the internalization of the virus into the cell. it has been reported that the binding of SARS-CoV-2 to ACE2 actually is very tight. We are talking about KD of 15 nanomolar. So this can indicate actually how challenging the targeting this protein is, because we are competing with a very tight type of binding. There has been several structural studies reported during the last 6 months or so, and one of them actually was by Rapp and his colleagues on Science. And it's known that this spike protein is formed by a trimeric assembly, and when it comes closer to the host cell, the receptor binding domain actually recognizes the ACE2 receptor, which is angiotensin converting enzyme 2, and then 2 steps of cleavage took place, and it helped the virus to be internalized. And these are the so-called hotspots, or the interface between the receptor binding domain of CoV-2, SARS-CoV-2, and the ACE2 inhibitor. And people already compared that with the receptor binding domain with SARS-1. And it's been concluded that there are additional interactions that help actually SARS-CoV-2 to bind in a tighter manner and much more easily, in a way, to ACE2 receptor on the surface of the host cell. And there are 2 conformations, as already been disclosed by several groups around the world, that that help really the viral infection ability of the SARS-CoV-2 to infect the host cell. There's a so-called open conformation and the closed conformation, and these are the amino acids involved in the closed conformation of the receptor binding domain of the S protein. And these are the amino acids also in blue that are engaged from the ACE2 subunit. The closed conformation is believed to significantly be involved in the pivotal role in signal transduction process or signaling cascade that helps in internalizing of the virus. Targeting non-structural proteins, which is the other major topic of medicinal chemists right now, one important or encouraging notion that those non-structural proteins actually are very conserved viral protein. And the other thing is those non-structural proteins have high similarity with other coronaviruses, especially with the SARS-CoV-1, the previous one. And right those days, there are more and more high-resolution PDB structures, which is to medicinal chemists actually a very good starting point. So that's why more and more groups are actually working worldwide on targeting those non-structural proteins. I will comment a little bit in some details, showing some structures on the proteolytic processing or the protealizing enzymes, the proteases. The one which is the chymotrypsin-like protease, 3CL Pro, or what is coined as or named as the main protease or protein, and the other is the papain-like protease. I have to say that the main protein or the 3CL Pro has been under intensive investigation by several groups, and hopefully we will be seeing some promising molecules as lead candidates to go into clinical, at least preclinical trials soon if the labs will be reopened again. when comparing the 3CL-Pro with that exists in SARS-CoV-1, the previous one and the current one, there's something like 96% identity in the sequence, and actually there are those 4% of differences occur on the surface of the protein and very rarely on the catalytic side. Actually, the catalytic side of the two of 3CL-Pro in both viruses is almost 100 similar, which actually blunt the rationale why people are using or trying to use the drug purposing strategy for identifying potential candidates to SARS-CoV-2. Again, there are some differences, but the catalytic sites are almost identical between SARS-CoV-1, the first SARS, and the current SARS. So there is a very good reasoning why to go for a drug repurposing. Those are representative structures of small molecules on this side, natural product, for example, and peptidomimic compounds that has been developed to target SARS-CoV the first, and now they are tried and examined and explored against the current SARS-CoV-2. And generally speaking, there are whether it's small molecule or natural product or a peptidomimic compound, the compounds are classified into non-covalent inhibitors and covalent inhibitors. And the covalent inhibitors actually, as shown in red here, have a reactive, all of them actually, each one of them must have a reactive functional group that binds covalently to the enzyme. And I will come into mechanism, how it is believed that, and why people are really focusing more and more on covalent binders or covalent inhibitors. I have to say that covalent inhibitors are also divided into two subclasses. One is irreversible covalent, so it binds covalently and stays there for a long time with T-half that is very long, and it's defined as irreversible, covalent irreversible, and the other type is covalent reversible. I will show one example of that. These are also another group, and I mentioned that because there are many and really wide, strange, and diverse structures of inhibitors that has been reported since 2003, since the first SARS-CoV-1 virus. And this group actually called the keto-amide group because it has a keto alpha to the amide group. This keto is close to the amide group. And this group is very promising in one hand. These are actually the sub-pockets in the binding side, the pockets where each group reside. But I mentioned that because one of them was used actually to get an X-ray structure of the protein, number 13B. And here you see the ligand, the inhibitor, actually residing, homing actually on the active site of the protein, on the enzyme. And not only that, that cysteine 145, which is one of the so-called dyade, the histidine close to this cysteine close to this histine are called the catalytic dyade. This cysteine 145 is the reason why people think of developing covalent inhibitors. It's bound to this peptidomimic compound via thiohemiketal, and this is a bond that is actually cleavable. So it forms a covalent bond, but it is weak enough to be reversible. That's a very good example of a covalent inhibitor that is reversible also. So this is a very informative structure, and people can, researchers worldwide are studying this type of binding, the conformational settings of the inhibitor inside the active site. And I just want to mention one more point, which is the ring in black, which is called the pyridone, which is a derivative of pyridine. So they are on one side alpha keto amide, but also they are pyridone-based compounds. So hopefully we will see more advancement based on those information. Another structure that has been reported very recently, which is the structure of main domain, of the main protein and protein with an anti-cancer drug that was used for colon cancer and other type of cancers for years. And this is the structure of the anti-cancer drug. And it turned to be that actually this cysteine 145 acts as a nucleophilic attack, a nucleophile, and it performs a nucleophilic attack on this urea bond, urea functional group, and it cleaves away the fluorouridine, leaving the enzymes labeled with, actually covalently labeled with the lipophilic tail. And I should mention that this previously, but that's okay to hint the different domains of the M protein that has been already identified. How does M protein in SARS-CoV-2 bind to Camphor or to another covalent binder, for example, like this Michael acceptor containing molecule or inhibitor? This cysteine, I don't know why it keeps jumping, but anyhow, this cysteine actually acts as a nucleophile, and it attacks this Michael acceptor. In this case, the adduct formed is actually a covalent, irreversible Michael. In other cases, for example, I have to show one of them. If there is a leaving group, like halo bromo compound, then there's no Michael addition, but there's really a covalent bonding. So the M protein can, through this cysteine 145, can either do or can perform Michael addition or nucleophilic attack or and replacement of a leaving group like alpha halo or bromo keto compounds. This brings me actually to the efforts to target RNA-dependent RNA polymerase. And the reason for that, because RNA-dependent RNA polymerase is very critical in duplicating the genomic RNA and the subgenomic RNA, and that are very needed to any duplication of the virus inside the host cell. And this has been characterized previously as a polymerase. And here we have the entrance of the nucleotide. So a polymerase actually uses a primer, and there is an elongation. But in order to do that, it has to bring in the certain complementary nucleotide. In this case, remdesivir actually resembles the adenosine that is needed originally for the synthesis of RNA. If in the region or in the area or in the pool around, remdesivir in the proper form is found, so the RNA-dependent RNA polymerase mistakenly misincorporate the remdesivir, which is adenosine triphosphate analog, instead of adenosine, ending up with actually cheated RNAs. It's not a normal natural RNA, it's an RNA that contains remdesivir instead of adenosine. And there is a phenomena called chain termination. So after 2 or 3 nucleotides, there is a termination of the RNA. So there's no full replication of the RNA. And if there's no full replication of the RNA, this can inhibit the viral replication. It turned out that remdesivir, which is GS5734, GS from Gilead Sciences, the company developed this compound or discovered this compound, is a prodrug of GS441524, this one. So the remdesivir is actually a prodrug. It's not the only prodrug that is used as a antiviral agent. Sofosbuvir, which is compound number 5 here, is also a prodrug, and tenofovir is also a prodrug. And I'll hint a little bit on later on the prodrug approach and why is that. All of them actually are nucleotide or /nucleoside derivatives, and they act as inhibitors of RNA-dependent RNA polymerase. I have to say that remdesivir was developed by Gilead Sciences for the treatment of Ebola virus and not for the treatment of the current pandemic SARS-CoV-2. But again, within the rationale that in the context of let's take up an existing drug and try it on SARS-CoV-2, remdesivir was brought to the stage, and it's been tried clinically for a while. And there are some reports that it has some effect, though with certain limitations. So remdesivir is actually a prodrug. It has to enter the host cell and undergo a sequence of conversions and cleavages, actually, in order to give the, sorry, I don't know why it keeps jumping, but in order to get this nucleoside monophosphate that has to undergo 3 additional 2 steps by kinases, intracellular kinases, to convert the monophosphate nucleoside to the triphosphate nucleoside. The minute this is formed, it can be a good substrate for the RNA-dependent RNA polymerase. And since it resembles adenosine, but it differs. Adenosine is purine, and this is actually a nucleobase with imidazotriazine scaffold. This is a CC bond and not a CN bond, and we have at one position C1 prime actually a cyanonet and nitrile group. So there are differences, structural differences that make of this enough different from adenosine. That's why when it is misincorporated, it ends in the so-called chain termination of the RNA synthesis. And if this happens, the virus will not have its full RNA for application or replication. Another prodrug, which is sofosbuvir, again, it is given as a prodrug. It has to be converted by cellular enzymes to the monophosphate, and then it has to be converted to the triphosphate form in order to be active. Okay, what we are doing, I mean, since 3 or more months, together with Hu Long and Abraham Carola Pudi, we decided to go and try, since my lab and their labs are closed, so we tried a so-called an effort to virtually identify some of spike protein interrupters. So this is a joint effort, and these are the aims. And the rationale actually to prevent the binding between spike protein and the ACE2 enzyme on the host cell surface, and by doing so, we hope to prevent the internalization of the virus. And the approach we adopted is a fragment-based approach, and it started with enamine protein interaction fragment screening this library, then scoring it, and ending up with a pharmacophore of 2 ligands and building a pharmacophore. Again, this was already explained. I will just go quickly over that. So the steps that we have been doing is this library has been screened by these two wonderful collaborators based on the PDB 6MOJ. Two fragments were identified and they were scored, and a pharmacophore was constructed. And then I came into the picture with my medicinal chemistry expertise, and we have a counteractive, almost, if not daily, weekly, between the computational team and the medicinal chemistry part of mine, in order to what I call have the human factor from the beginning. These are the two fragments that have been identified, fragment 1 and fragment 2. These are in ChemDraw, and these are the parts of each fragment. Fragment 1 is actually a derivative of pyridone, methylpyridone, and dihydrobenzofuran. Fragment 2 is a derivative of phenylacetamide on one side in red, and dihydroimidazopyrazine in the other side. So we decided actually to dock those two ligands, and they were docked. And it turned out that those two ligands occupy a similar binding pocket on the receptor binding domain, very close to tyrosine 505 site. From here on was actually a process of fragment growth, merging, and diversification. And what the chart here actually explains just view of what has been done already. So the idea was, if those two ligands occupy a similar binding pocket, then let's fuse them and have them connected covalently, and replacing either the pyridone or the phenyl, one with the other, of that region. And this was done on this side or in this side. We ended up actually with growing ligands all the time. So on the right hand or the left hand, it depends where we link the two parts together. And if we go downstream, actually, again, replacing the two rings in violet with a phenyl ring, and keeping the hydrogen bond donor acceptor with by having a phenol. So we end up with another small library of compounds that are phenol derivative, tri-substituted ones. And I'll show just few examples of the top hits that we have been working on for the last 3 months or so. This is a collaborative effort. So we have the proposed library. We started with the enamine fragment library. Then we have the two ligands. There is we defined the pharmacophore, then modeling and docking, scoring, and then the medicinal chemistry input. And then I propose another library that is totally different, and this goes in a collaborative circle of work. Merging those two ligands, we end up with a third ligand, and this, as shown here, the new, what we call it 000151, which is hit 1. As we see, the phenol ring in the center is really occupying the same binding site, and the tri-substituted, the other groups are actually, it's like a triangle around this phenol core. And these are the amino that are participating in the interaction with this hit 1. The idea was to go from hit 1 to try to improve it. And the way to do that is to look into, again, this is hit one. As we see, the phenol is actually on the top of the narrow binding site that is behind it. We ended up with, again, trying to improve hit one, we ended up with hit number 2. And the idea always, and this is only just part of the considerations, I looked into the binding site, trying to identify close to tyrosine 505 what other amino acids are in the vicinity and what amino acids can contribute in a complementary way to the ligand. So in other way, what functional groups I have to add to this hit number 1 in order to get a better interaction with the receptor binding domain. That is, this binding site close adjacent to tyrosine 505. And for example, I converted, for example, the amide, the acetamide part of the fusion of the two ligands and converted to oxazole ring. And what happened is, as we see here, the oxazole ring is actually occupying this binding pocket, this narrow binding pocket. The next step was to look around and to see how to improve, because the scoring was around 7, this hit number 2. So I focused on 2 parts at this stage. One is to take this oxazole ring and try to explore and modify there. Maybe we can extend it or exploit the polyaromatic cluster that exists in this side of the binding pocket, on the behind this pocket, or on the other side, I focused on the trifluoromethyl which is this part, and try to see what amino acids are on the vicinity there and add functional groups that can interact with those amino acids. So, and these are just examples that has been done. This is the, again, hit number 1 with the oxazole. You see the oxazole is inside the pocket, and the phenol ring actually is on the top, as shown in the dotted circle, oval here. I added in one exercise or one trial methoxyphenyl to the oxazole, so I end up with phenyl oxazole with a methoxy on this position, and this was among something like 150 compounds, again with a score of almost 7. But as you see, we are no more inside this pocket. The phenyl oxazole actually is residing outside the pocket, but the phenol in the center is still almost in the same position. In a following step, I took hit number 2 and focused again on the thiophane part, on this part, and tried to see if I can introduce functional groups that can interact with glutamate 484 residing on this side. And by adding an amidine group on cyclopentanone, I ended up with hit number 3. Again, it binds to the region, the same region almost, but the ligand actually is shifted. All the ligand is shifted. It seems that due to the strong binding between the amidine group here and the glutamate 848, the whole ligand, which is in yellow, is shifted away. These are the top 9 hits. This is number 1. And without going very deeply into the chemistry, because I don't realize who really listening to me, but I can hint into 3 points. One if we keep the dimethoxyphenyloxazole, which is in red, constant, and the indole ethyl as constant, and the originally identified imidazopyrazine, dihydropyrazine, as constant, it seems that the cyclopentathiophane ligand has been identified as one ligand. If we attach to it amidine, it can bind tighter to the same binding.
Excuse me, sir.
Yes.
Can you increase your volume, please? And we have a time constraint for the next talk to come.
Okay, so to increase my voice or decrease?
Increase the voice.
Oh, increase the voice. Okay, I will try. I'm using. So I'll try. Is it better now?
Because this is just from the YouTube viewers, and
okay. So I hope
it's
it's better now.
Yes, sir. And
okay.
And sir, so we have a time constraint and we have some questions to ask as well.
Sure.
Before closing the session. And yes, sir.
Can you understand? Okay. Yeah. Thank
yes, sir. Thank you.
Okay. So again, we are in this stage of having hit number 3, but it is displaced from the original binding packet that we aimed at. So I'm in a phase of thinking how to solve it and bring the ligand back to the tyrosine 505 and improve the scoring. Still, the scoring is around 7, so we have still a way to go. But this is again an effort that has been done in a collaborative way between India, Palestine, and the United States. And I should thank every one of them for that. Where are we going from here? So I'm planning to virtually optimize in cooperation to get an improved hit, and hopefully with drug likeness properties. Then if the labs will resume, hopefully we will go to synthetic stage to synthesize the predicted ligands, and then to go for in vitro. And there's someone within the COVID-19 cluster, some counter partners who are ready to test the minute we get, hopefully, the compound. With Abraham, we have been doing the same, almost similar approach, but on non-structural protein 15. And I won't say more because, again, of time limits. And by that, I would like to thank you very, very much for your attention. And I'm ready to ask, if I can, any question you have. Thank you very much.